Okay, for the B cells activation, as we said, the B cells is part of the humoral immune response. What does it mean? It means that it acts on the non-self antigen through the fluids of the body. So the B cells doesn't need contact with the antigen as we saw in the T cells. So how does it do this? By releasing antibodies. Those antibodies will go and attach and neutralize the non-self antigens. Antibodies, the first thing we need to understand it is what are those antibodies? What is the antibody? So the antibody is technically a group of proteins that they are sitting in a certain format like this, as you can see, like letter Y. We have in the antibodies like two regions. We have the constant region and the variable region. Okay, those are made out of heavy chain and light chain. This is related to the nature of the amino acids that forming the antibody. Now you don't need really to care too much about the light chain and the heavy chain, even the constant and the variable region. What I need you to understand, call it variable region, because this is the area where it's different and between antibodies. So most of the antibodies, if not all of them, will have the lower part as the same. The only changing part is this part, the variable, and that is where the antigens are going to bind. Okay, so this in our drawing, in our symbol drawing, it's this region. So that's why sometimes we draw it like this, sometimes we'll draw it like this, sometimes we'll draw it like this. Okay, because this is the variable region. The relationship between the antigen and the antibody is a 3D conformation. They need to fit to each other, otherwise we will not get a recognition of the antibody, or the antibody will not be able to bind to the antigen. Okay, so that's the variable region. So, and this is the general structure of the antibody. Now the antibodies, we have 5 types or 5 major classes. The first one is the IgM, and this IgM is pentamer. What does it mean? It means it's like there are 5 antibodies connected to each other, and therefore each one of those antibodies can bind one cell or different antigen. Okay, and that's why we saw it in the blood. There is agglutination, because one antibody like IgM can bind here in this example or in this image, like theoretically it can bind 5 different cells. And imagine you have like 100 of those. Each one can bind 5 different cells, and you will have the same cell bound to 2 IgMs, and this IgM is binding a 3rd cell, and so on. So that's why we get the agglutination. For the IgM, the main thing about it, that it is the largest in the body, the largest antibody in the body, and this is usually the first antibody during the primary response. This is the first antibody that is get released during a primary response. Primary response, it's like the first infection. What do I mean by first infection? Like the first time this pathogen gets into the body. Think of a newborn baby get the flu virus, which is something dangerous then, but just to understand what does it mean, like the first infection. Or even an adult, each year we have a different strain of flu, and let's assume that this year we have strain alpha 1 beta 3, for example, of the flu virus, and this is a new strain. So if this virus get into this individual, that is the primary infection or the first infection, and therefore we will have a primary response. Okay, so this IgM is the first one that usually gets released in response to a primary response, and this is like very effective against bacteria. The IgM is very effective against bacteria. So this is the first antibody. The second antibody is the IgG, and this is monomer. What does it mean, monomer? It's just one antibody, and this IgG is usually one of the late antibodies. So we will see it toward the end of the infection, so late primary response. But on the secondary infection, this is the primary one. This is the major one. What do I mean by secondary infection? By the secondary response, that's like the second time infection. The same strain of flu came back to that individual. The IgG is the one responsible for the hemolytic newborn disease. Okay, the one that the mismatch between the mom and the fetus. So the IgG can cross to the placenta, and therefore it can attack the fetal circulation. But of course, as IgG will help also in protection, so it will provide some immunity to the newborn babies. This IgG also can leave the bloodstream and go to between the cells. So if there is an infection there, it can go between the into the extracellular fluid. Then the third one is the IgA. IgA is a dimer. It means 2 antibodies connected to each other. This is the only antibody that can go to the body surfaces. Like it can go to the mucus of the nose, for example. or mucus of the mouth, or even to the skin. This IgA can go through the breast, through the milk. If a mom is breastfeeding, this can go with the milk to baby and therefore provide immunity for the baby. And then the fourth one, we have the IgE. IgE is also monomer. This is usually associated with allergies. So this, as concentration is very low, but it will spike if there is an allergy. Think, for example, like to peanuts, for example. So there are like peanut specific IgE that will get released in the stream, in the blood, because of someone eating peanuts or coming with peanuts, sometimes even smell. So IgE is usually associated with allergies. Then the last one is IgD. Also, it's a monomer, but the thing about this IgD, it's fixed. It cannot be secreted. All of the other ones, all of the other four can be secreted. This one cannot be secreted. And therefore, this, we'll call it the B cell receptor. So this would remain on the surface of the B cell. So it's IgD. It will remain always on the surface of the cell. Okay, how do we now activate the B cells? So we have in the lymph nodes different types of the B cells. Those different types are displaying different IgDs. So all of those in the drawing are IgDs. But those B cells, okay, those are mature, not yet activated. Each one of their IgD is showing a different 3D shape. This one would show like this. This one like half circle. This is a flipped triangle. And let's say this is like a square. And the antigen we have is a triangle. This cell cannot recognize this antigen. Why? Because its antibody is a circle. So this would recognize the circular antigens. In this example, it will not recognize it. This one is the same. It will not recognize it. If the antigen fits here, it doesn't fit. So we won't get activation. Only this one will get activation. Why? Because it fits perfectly on its IgD. So what does this mean? It means that in the body, in the lymph nodes, those B cells, we will have like a huge number of thousands of those B cells, that each one is specifically designed for a specific antigen. Once this antigen, non-self antigen, get attached to the B cells would start its activation. The first step in activation is forming clones. It will form clones. It means it will start to divide. And this division would keep going several rounds. Eventually, we will have two types of cells. We will have plasma cells from the activated B cells. We will have plasma cells. Those plasma cells are the one responsible for secreting the antibodies. The same antibody as here, because this is the one going to catch those little triangles. So the majority, the bulk of the activated cells or B cells are going to give us plasma cells. Then a few number of them will give us memory B cells. So those memory B cells like a record, like a log for the infection. And that will be the primary response. In case we get a second infection, then those B cells are ready to clone immediately. They will start to divide and give us plasma cells. So there is no need for reactivating them because already they are active. They just need the signal to start dividing again. On the secondary infection, again, we will have plasma cells. Those are going to secrete the antibodies, and we will have also memory B cells that would remain in the body in case there is a third time infection. Now, what is the advantage of this memory cell? As we said, it's going to respond in case we have the second infection. And in fact, it's going to give better response, a stronger response in a shorter time comparing to the primary response. So see here, for example, the primary response in time, it might take like 7 to 10 days to peak in the concentration of the antibodies. And this peak is not really significant. But on the second response, we will have significant increase in the antibodies production in shorter period of time. This might take 3 to 5 days to peak. So that is the secondary immune responses. And that is the idea of the vaccines. Okay, the vaccines are usually a step to establish a memory B cells. So we will introduce a form of the pathogen. Maybe it could be the shell of the virus, just the shell, or maybe it could be part of the shell of the virus or part of the bacteria. We'll introduce it to the body, get a primary immune response, keep the memory cells. In case this infection came back, we already have advantage over it. So we will get better immune response in shorter period of time. Now, sometimes the B cells will bind an antigen, but they are not sure about it. They need a confirmation on it. So that is when they will turn into the T helper cells. The T helper cells, as we saw earlier, will bind to the antigen. would recognize the MHC2 and they will release cytokines. Those cytokines will tell the B cells, yes, this is a non-self antigen. You need to get activated. Or they will decide, no, this is benign. This is a self antigen. You don't need to get activated. Okay? So sometimes the B cell activation would involve the helper T cells. That's why the helper T cells are extremely important immune cells in the body, although they are not directly involved in dealing with the antigens, meaning that they don't kill the cells, they don't develop, they don't divide, and so on. But again, they will mount or they will regulate this immune response. Okay, now after understanding all of this, let's try to talk about the active and passive immunity and what does it mean. Now we are talking about active and passive immunity. What does it mean, active immunity? It means that the pathogen infected the body and therefore we got immune response. Like a normal infection, flu virus, for example, or bacterial infection, that is active. The passive, it means there is no pathogen entered the body, but there are antibodies in the system. Okay? And we'll understand this. Now, both types, active and passive, will have two subtypes. So we will have active immunity and passive immunity, active and passive, and both will have either naturally acquired or artificially acquired. Same thing for the passive, either naturally acquired or artificially acquired. So what is an active naturally acquired? It means, as we said, active, it means the pathogen got into the body, right? So this is our classical example is the flu virus in body. When you get the flu, for example, or bacterial infection. Artificially acquired is the vaccine. So here we inject the pathogen or a form of it and therefore develop a memory cells. That is the artificially acquired. The passive naturally acquired, it means that the antibodies entered the body. Like what? Like the antibody we talked about during breastfeeding, the IgA. IgA moves into baby through milk. So the baby did not see the pathogen that caused IgA to be secreted. Maybe it was bacterial infection because of a cut in the mom's finger. Okay, and her body developed IgA, right? This IgA would go to the baby through milk. So the baby did not see that bacteria, but in her system, in the baby's system, there are the IgA antibodies. Artificially acquired, it's when we introduce the antibodies introduced to patients, for example. Like what? In some treatments of cancer. In some treatments, they will call them monoclonal antibodies. So here they inject the patients with antibodies that have been developed against this specific type of cancer, and those antibodies will go and kill the cancer cell. So those are the two types and the subtypes of active and passive immunity. This is the video about the entire immune system. Please watch it. It's very useful. It will show you animation of all of the things that we talked about. The last thing we will talk about in this chapter are the immune deficiencies. And here we have two big diseases. The first one is the AIDS. AIDS is the result of the HIV infection, human immunodeficiency virus. And the problem with this virus that it was targeting the helper T cells. That's why at the beginning it was really difficult to deal with it, to understand how does it do it, or that's why it caused like immune deficiencies, because it's killing the T cells. So the HIV virus used the CD4 to get into the cells and therefore destroyed them. Now HIV is not as, or AIDS in general is not as severe disease, or a disease that would cause problems, significant amount of problems in the patients. Patients can live now with AIDS for extended period of time, and the treatments now are better in dealing with those viruses. Then there is the autoimmune disease, and this is the big group of diseases that can virtually target almost any organ in the body. So we'll have the joints, arthritis, we have the muscles, aches, we have ulcers in the mouth and the nose, ulcers in the digestive system, we have infections in the lungs, we have infections in the hearts, we have autoimmune diseases that will target the fingers and toes, and therefore those are always cold, and so on. The pancreas, diabetes mellitus, now it's believed like autoimmune disease. So there are plenty of those autoimmune diseases. In all of them, the concept is the same, that the T cells and the B cells are attacking self cells and therefore destroying them. Why? Nobody really knows about them, So this is like still yet to be discovered, the reason why those cells attack the self-antigens. The last one is the hypersensitivities. It means that the body is more sensitive beyond normal levels. The problem with this, it will lead to inflammation, and always, even with the small type of infection, sometimes the immune system will exaggerate in its responses. Hypersensitivities, there are 4 subtypes depending on the cause and depending on the involvement of the cell, depending on the degree of inflammation. We will have type 1, type 2, type 3, and type 4. In general, what I need you to understand that the hypersensitivities, that's the body would respond in exaggerated way comparing to other patients. Okay, you don't need to care really too much about the different types of the hypersensitivity. And that's it for this chapter. Let me know if you have any questions.